the whole reason why TCR T cells and TCR therapies are going to be effective in the solid tumor space is because they can access these targets that are not cell surface proteins. Because it turns out there are very few cell surface proteins that are completely specific to cancer.
I may be talking about the peak sales. It was like a $400 million figure for the annual sales, and with pretty high gross margin as well. But then I saw some figures from the analysts from Guggenheim that they projected a bit lower, maybe 180 million. What's your take on this?
Don't comment on the analyst's forecast for obvious reasons. I talk about what we believe.
Do you prefer Philly Cheesesteak or Oxford Fish and Chips?
Philly Cheesesteak, I'm fine, sorry. I guess.
Bienvenue to a new episode. I'm your host, Philip, and on this show I'm interviewing the best Europeans in biotech. Help you grow. Cell therapy, especially CAR-T, has changed how liquid tumors have been treated, but solid tumors were harder not to crack. In August this year, the first engineered cell therapy was approved for a rare form of sarcoma. It's a TCR-based therapy developed by Oxford-headquartered company Adaptimmune. So I talked online with the CEO, Adrian, who lives most of the time in Philadelphia. Adrian has been with the company for almost a decade. He joined as CFO before transitioning to CEO in 2019. I don't know him personally, but I followed Adaptimmune since almost 10 years and knew some of the other C-levels, and I've heard many great things about Adrian. We talked about the commercial rollout of Tecelra. We also talked about what's next in cell therapy, as well as Philly cheesesteak versus fish and chips. So, here's the conversation with Adrian, and please hit the like and follow button if you're enjoying it. Thank you to today's sponsors, Merck, for accelerating therapeutic antibody development. Whether you are identifying new targets or optimizing your production, Merck has you covered. They have a comprehensive suite of tools from functional genomic screening to high-throughput purification. With cutting-edge advances in B-cell, cloning, and hybridoma development, you will both save time and resources. Merck is also a proud partner of today's guest, Adaptimmune. So, want to learn more how Merck can be your partner for therapeutics development? Click on the link below to download your brochure and get one of these cool antibody pins. All right, Adrian, welcome to the show.
Hi, great to be here.
I wanna start with the commercial rollout. Obviously, you got Tecelra approved for patients was in August. Great news. So yeah, how is it going with the patient rollout?
So Tecelra approved very beginning of August, and that's a sort of fairly big transition for companies like us. But actually, in terms of the relationships with the sites and the places that are going to be actually making Tecelra available to patients, it's really an extension of the relationships that we've had with them for the last decade or so, because we've been developing the cell therapies for sarcoma, synovial sarcoma specifically, and other soft tissue sarcomas for at least 10 years now. And so it's more sort of like it's an evolution of that, but it's a really important evolution. And now for the first time, we've got to be using the commercial diagnostics to be able to test the patients. We've got to get the treatment sites up and running. We've got to get the patients flowing through those sites, and all of that has sort of happened at pace. There's been an immense amount of enthusiasm from the sites, because this is the first therapy, the first new therapy that these physicians have had available for their patients for over a decade, and so they're really enthusiastic. We've had a great uptake in terms of getting sites active, patients coming through the system, and we've sort of said it's going to take 3 to 4 months to get the first patients all the way through that process, to get them treated. And we anticipate doing that by the end of the year. So we are deep in the bowels of it at the moment.
That's amazing. I was about to ask how many patients are already enrolled or already went through the process, if that was disclosed.
Yeah, we haven't disclosed that. All that we've disclosed is that we just want to make sure that everybody's realistic about the time frame it takes to get here. I think there's been a couple of launches in the cell therapy space where that time frame has been a surprise to people. But it takes time to get patients tested, to get the reimbursement sorted out, then to get the patients apheresed, and then treated. So we're in that process. We will dose our first patients this year, we anticipate.
That's amazing. And actually, you mentioned on the reimbursement, I saw the pricing. I think per dose, the listed pricing is $720K, if it's correct.
Yeah.
Yes.
Yeah.
Like, I mean, just to put it into the context as well, it seemed when I saw the figure, I was like, oh, that seems pretty high, even compared to, let's say, a CAR T, which is already, I think, more than 350 and already pretty high. Can you why what's the reasons behind?
Yeah. So the CAR Ts now in the US are priced, most of them are priced between 500 and 600,000. It's a modest premium to the CAR T therapies that are out there, and I think reflecting a couple of things. One is the absolutely spectacular data that from the Spearhead 1 trial that was the basis of the approval, which in this space, it's difficult to overstate how transformational it is.
I agree. I saw the data published in Lancet. I was like, wow, that's amazing.
Amazing data, and for this patient population, that's very small. I mean, this is a very targeted therapy. It's targeted on patients who have the right HLA type, right tissue type, and to have the right target. So it's a discrete ultra orphan patient population with transformational therapy, and that's the basis of the pricing that we rolled out and we anticipate over the coming years.
It's right. It's around 1,000 patients per year in the US alone. Right?
Yeah. So there's about 1,000 to 1,300 patients who are likely to be diagnosed with synovial sarcoma each year. So synovial sarcoma is about 10%, up to 10% of soft tissue sarcomas. So it's about 13,000 soft tissue sarcomas. About 1,300 of those are synovial sarcomas. We target about a third of those, so maybe 400 patients a year in the United States, and then probably a similar number, maybe a bit more in Europe, and it's tissue type, it's HLA-A02, and its target type is MAGE-A4. So you got to be biomarkers for both of those. So it's very focused therapy.
I think, that's also I think one of the limits of TCR therapies in general. I remember talking with Baiji at Immunocore and Happy at Ematics. They were both on the show, and they also had to exactly have the right HLA that is presenting the right peptide for the TCR to recognize, and has to be super specific. Can you talk a bit if you agree, and expand a bit on that if that's really a limit and how you overcome it?
Yes, so I think about this as actually as the potential and the opportunity. The whole reason why TCR T cells and TCR therapies are going to be effective in the solid tumor space is because they can access these targets that are not cell surface proteins. Because it turns out there are very few cell surface proteins that are completely specific to cancer. So even in the CAR T space, where the CAR Ts target cell surface proteins, they're not targeting cell surface proteins specific to cancer. They're just targeting lineage proteins for a B cell, on the grounds that if you have a B cell malignancy, I can clear out that lineage of B cells and cure your B cell malignancy because you don't have any more B cells.
CD19 is not the...
CD19, for example. No, it's not specific to cancer. It says this is this type of B cell, and that therefore, if I get rid of all of those, I've got a pretty good chance of getting rid of your cancer. But I also get rid of all your B cells. Yeah. But that doesn't work very well and you can live without your B cells, and you regrow your B cells, and hopefully they grow back without cancer. But for other cancers, like if you take lung cancer, if you said, well, I'm going to get rid of all your lung tissue in order to get rid of your lung cancer, that's unlikely to be an effective treatment strategy, and that's true. Now, there are places where you could say, well, that is an effective trade-off. Prostate cancer, for example, you could say could be an effective trade-off. There are some places like breast cancer, but actually finding targets that are really specific to cancer is quite difficult. And there are very few cell surface proteins. And when you talk cell therapy, you've got a different level of selectivity that is required, because for many other types of therapies, including bispecifics, like Immunocore's bispecific, the bispecifics, you can tolerate a certain amount of off-cancer on-target activity. So you can have a certain amount of expression in normal tissue and still have a therapeutic benefit, a therapeutic index. Mm-hmm. But with cells, that's very different, because the reason is that for most other therapies, when the therapy is most effective is immediately after it's been given. And then it tails off. Your sort of classic PK/PD sort of, the effectiveness of the therapy tails off over hours if it's a small molecule, days, months if it's a protein. Yeah. But with a cell therapy, when the cells see target, when they engage with target, the first thing they do is multiply. Yeah. So they kill what they see, and then they divide and they multiply. So you get this expansion against whatever it's seeing.
So sometimes they become too active.
Sometimes they become too active. And if you have even low levels of target expression in normal tissue and the T cells see them, they will attack that normal tissue. So what it means is that you need very, very clean targets to go after with a T cell. And most of the things that you would normally target with cancer therapeutics are not sufficiently clean. And very few of the cell surface proteins are sufficiently clean. So you've got to go inside the cell. Yeah. Now, the problem is you can't get T cells inside the cells. So you've got to rely on the fact that the cell protein machinery is constantly breaking down proteins that are inside the cells, venting them up using HLA on the surface of the cells. And that's what T cells actually see. They see this presentation, the context of HLA on the top of the cells. So I think about HLA as the way in which we get access to the intracellular targets, the intracellular proteome. And there are many more proteins that are specific to cancer inside the cell than there are on the cell surface. So actually, whilst it is a restriction, it's a mechanistic restriction. It's down to the underlying biology of and it's actually the way in which you get access in a selective fashion to these cancer targets that are very difficult to target otherwise. And then the other thing that I would say is, most of us who've been developing these have been developing them in the context of HLA-O2. And that makes sense from that's where the largest population is. Okay. But there are other HLAs and they present slightly different peptides. And so you can expand the opportunity once you know that the target works and that cell therapies against that target work. You can expand it to other HLA types. And overall, if you had sort of 3 or 4 HLAs for each target, then you would end up with 60-70% coverage, maybe up to 80%, depending on the geography of the world population. So it is viable to develop broad-based therapies that could treat the vast majority of people with relatively small numbers of HLAs.
Oh, that's the better. Yeah. Yeah, I remember talking with Harpreet. He was making that point. It was not clear to me that the TCRs were not recognizing they always talk, we target the intracellular targets, but it's presented on the HLA and it's very small piece of the peptide. So you need like ultra-specific, bispecific or TCR engineered cells that are ultra-specific to recognize.
Yes.
Now everything makes sense. Okay, that's good. And maybe to get back to patient access, also, how is it going in Europe?
So we've had interactions with the regulatory authorities. We conducted some of our clinical trials in Europe, and we're consolidating the information from the Spearhead 1 trial, which had multiple cohorts, some of which is going to be required for confirmatory evidence in the United States. And the totality of that, we believe, will be able to be submitted in Europe in the years to come. So it will come behind the United States. But there is a population of patients in Europe that I think is addressable by Tecelra, and over time we will be able to address that. I think also got to think about this from a business context, in the context of other things coming through, and the infrastructure needed to support European launch. And we could think about that ourselves, or we could think about that in partnership as well.
Yeah. Okay. Because you didn't have a fast track mechanism for Europe, or you didn't apply for it.
We have prime designation in Europe. And the discussions with the agency were conducted under that designation. And so we believe we understand what will be needed to receive approval. I think the commercialization of cell therapies is more challenging in Europe than it is in the United States, but all the CAR Ts have shown that it is possible to do that.
Okay. Yeah, no, that's good. Yeah. I mean, I think it's an amazing option for patients, so should be available also to more globally. Yeah. Maybe talking about the peak sales, like I was curious. I heard you talk on the Zach's show podcast interview about the projections you had, which were, I think, you talked about the whole sarcoma franchise, maybe not specifically to Tecelra, but I remember, and I wrote it down, was like a $400 million figure for the annual sales, and with pretty high gross margin as well. But then I saw that at least some figures from the analysts from Guggenheim that they projected a bit lower, maybe 180 million or something. At least that's the number I saw. Why? Like, I mean, it seems like a 1 to 2 difference. Like, what do you do? Like, what's your take on this?
Yeah, I can't comment on the analysts, on the specific analysts' forecast for obvious reasons. I talk about what we believe. I mean, so there's 2 products. Tecelra, the first approved, and then behind that is a product called Leticel at the moment. It'll have a brand name in due course. And Leticel, so Tecelra targets MAGE-A4, Leticel targets NY-ESO. Tecelra is for synovial sarcoma, Leticel is for synovial plus and also for myxoid sarcoma. Leticel has completed its pivotal trial. The final data for that we anticipate at CTOS this year, in mid-November. We've already talked about the interim analysis, which is a little old, but also showed that trial had met its primary endpoint even at the interim analysis. So this is going to be a positive trial. Therefore, we believe that we will be able to register that product and have that on the market in about 2 years' time. So you'll have Tecelra and Leticel coming side by side. And the beautiful thing from our perspective is they go through exactly the same commercial channels and footprint. So the commercial channel that we're building for Tecelra can just absorb
same treatment centers.
Okay. Same physician. It's same systems of supplying it, although the manufacturing process is different, very slightly different. We think there is a sarcoma franchise with massive leverage from a business sense in the sort of commercial channel that we're going through and the footprint we're building. And because of that, we're looking at, okay, what's the value of that to us? And the 400 million number that we put out there was the value, I think, in the United States alone of Leticel and Tecelra, peak year sales in the launch indications alone. So we think that's and we made that available because if you go back to last year, I think people thought that the entire sarcoma business could be worth something like $80 million peak. And so we thought, hang on, you're just in completely the wrong place. One, you got one product, whereas we've got two. The pricing is not where we have now sort of landed on. 3, we think you're underestimating the patients, etc. So there's a whole sort of set of stuff there as to why we wanted to rebase where we are. But we think about that as a good start. We can address that. We can address that at a decent margin, and then as we do that, we can also be thinking about how to expand that. Once we're commercially invested in this space, are there other soft tissue sarcomas that express MAGE-A4 and NY-ESO, and how do we go after those? Could we think about use in earlier lines of therapy? Can we think about geographic expansion? And then how can we bring the rest of our pipeline through in sarcoma so we really own this sarcoma space for cell therapy?
That's good. And I can imagine from a business strategy or corporate strategy, once now you're commercial, but once you're profitable and commercial, it's a very different game, and you can have much more flexibility, investment, expansion, etc., etc.
Very, very much so. And I also think there's a big difference between the investment in the R&D to support the expansion of an existing franchise with existing products versus the investment in R&D in things that have yet to demonstrate that they are effective or can effectively generate sales. And just the risk profile of that, of saying, well, okay, I'm in this space. What does it take to expand the label into this space? The risk profile and the investment profile of that, it just looks very different. And there's a different return on investment approach to that. And once you've got products, if you look at the history of successful biotech companies, and a lot of them have been successful by finding something that works and then working out how to maximize the benefit that product can provide to patients on a broadest possible front.
Yeah, I'm thinking about Allogeneic in Europe, which have built a massive franchise on basically a single product. Like it's pretty. It's not so therapy, but it's an amazing example.
But it is also true of some really big companies. I mean take Keytruda, take Merck and Keytruda. When they initially had this, they didn't really realize what they'd got. When it was first being developed, they were like, well, I'm not really sure how big. It's the largest product in the world. Or take some of the
It's crazy on a single.
Indeed, yeah.
I mean, basically single indication almost, and it's crazy.
Yeah.
Yeah. So I guess from a next step also, like 2025 will be a key year for you guys, I guess, to show that you can reach the sales you have kind of predicted, and that you have also the profit margin that follows, and that you're on the right track to show all the predictions I can imagine.
Yeah, absolutely. And I think, whilst we said we anticipate to have our first patients treated and first commercial sales in 2024, the real sales are 2025 onwards, and that's when we'll see the patient flow through and the delivery. And then the other piece of that is also moving that second product, lete-cel, forward as well and behind it, so that we've got the confidence we can get to the other patients that that are addressable, particularly in the myxoid sarcoma space.
Yeah, that's great. Maybe, we talked about the current topics, and the next step, maybe to zoom out a bit on the history of Adaptimmune and where you joined where you came into the picture. Because I think the history from what I know from Adaptimmune is really it's very interesting from where there's some science coming from Germany, from Medellin. Baby cousin was the Immunocore, where you split it, cell therapy versus bispecifics, where it's very European-led, cross-continent, but with a very global development. And you joined almost 10 years ago. I guess you will celebrate your decade soon, I guess.
Yes. Early next year.
And you joined, I think, as CFO first, then you transitioned to CEO. So can you, maybe start with the history of Adaptimmune, maybe before you joined, and how then it went on when you joined until now?
Yeah, sure. So Adaptimmune itself, as Adaptimmune, has been around since 2008, when it spun out from Medellin. But the history goes back to about the year 2000, when when technology was spun out from the University of Oxford, or licensed from the University of Oxford, into a company called Avidex back in the history, and that Avidex was really the precursor of both Adaptimmune and Immunocore. So Avidex got acquired by Medigene, and then separately Adaptimmune and Immunocore spun out from Medigene in 2008, about 3 months apart from each other, had access to the same fundamental intellectual property, which is how to engineer the T cell receptors, two fundamental tools for engineering T cell receptors. And obviously Immunocore uses that for bispecifics, which requires a very different form of engineering to the engineering that you do for a cell therapy. So from 2008 until 2014, Adaptimmune was financed by high net worth individuals in the Oxford area, and was researching how to incorporate T cell receptors into cells, and moving forward into the clinic in very small trials in the United States, in partnership with leading academic centers, including University of Pennsylvania.
There are no VCs involved in the early days. Unless
not traditional VCs. 2014, they did their first VC round, proper VC round, led by NEA and OrbiMed, and that put us on course for an IPO in 2015. And this was when cell therapy was sort of starting to become really super hot. You just had the first CAR T data. Novartis had done its deal with Carl June a few years before, and it was clear that CAR Ts were going to be huge, and the proposition that CAR Ts were maybe not going to be the solution for all cancers, and that TCR T cells would be another wing of that, was sort of starting to become accepted. And so first round of financing in 14, IPO in 15. I joined in between those two as CFO in order to help take the company public, which we did very successfully in 2015, in May 2015. And what we did with that is we then sort of used the proceeds and the momentum that we gained from those financing and from the belief in cell therapy to really invest in some of the core components that we believed are going to be critical for cell therapy, principally manufacturing, and our manufacturing internal manufacturing for our T cells, and so the history of that through then to the approval of Tecelra earlier this year is sort of a focus on moving from a technology platform company into a cell therapy company with the capabilities that you need to actually design, develop, and deliver, as we say, cell therapies, which we think are really specific, poorly understood by the rest of the world, and quite different to my experience with small molecules and biopharmaceuticals when I was my predecessor company, GSK.
Yeah. Oh, that's amazing. And then between the IPO to now, like, and your transition to CEO, how did that go? Like,
transition, I think we're well. So I transitioned in 2019. At that point, we'd seen the first signal in sarcoma with what is now Tecelra, as then called afami-cel. Lete-cel was being developed actually by our partner GSK at the time, and we were sort of moving the rest of our pipeline into the clinic. Took over.
How you got in via GSK, or you knew the company via GSK, or there's no connection?
No, I did. So I ran business development for GSK for a number of years, but actually when they did the deal with Adaptimmune, I was CFO of the North American business at the time, and so I had no understanding of Adaptimmune. I knew Avidex from way back when I'd interacted with them. But I did not know Adaptimmune at all. Until I sort of got the call out of the blue about this small biotech company that was going public.
It's crazy sometimes in biotech. The biotech world is so small, but sometimes also you sit very next to something or an opportunity and you don't see it.
No idea that was happening. And you can imagine, as the commercial organization of GSK, the focus is not on, there's a really interesting proof of concept in humans study going on in this R&D organization. There's X years from the market. You're very focused on the respiratory franchise, the vaccines franchise, and the stuff that's actually real time. You can sit right next to it and not know what's going on.
It's amazing. That's cool. And then maybe you talked a bit before about I'm curious of the skills or what you brought from your experience at GSK. Because, you said it's very different to expand the label in small molecule, but at the same time you have this commercial exposure and what it takes. So can you elaborate a bit on that? Of I don't know, what was the most useful skills or most useful experience, or some examples you had where it was very valuable?
Yeah. So I'm not a scientist by training. My first degree was mostly molecular biology and biochemistry, but I'm not a scientist by training. But I had a number of roles at GSK, and actually probably the most useful ones were the ones that sit on the interface of the science and the business. So I spent a lot of time in their business development organizations of various types, ultimately leading worldwide business development for a number of years. I also ran their venture capital fund, SR1, for years. And the beauty of those roles is that you get to look at an awful lot of science and R&D projects. I probably did diligence on, whilst I was running BD, maybe we did diligence on a couple of hundred companies, a couple of hundred programs over that period. Deep diligence. So you get to look at, and when you do diligence on a development program, it's a fascinating thing because you get to see in one go the longitudinal history of all of the development decisions that were taken all the way back into research, and you get that nicely packaged up, and you get, why did you take that decision, and why did you choose that trial and not that trial, and that indication and not that indication, and I think that was incredibly instructive in how good R&D gets done, and then also the overall what to pay attention to. I'm fond of talking about that. I didn't look when you're running a business development organization. You've got to trust that the experts in the company that are doing the diligence are doing a good job. And so by and large, I did not look at primary materials for most of the stuff that was coming through diligence.
But you have to understand. You have to, understand them, and
Yes, you do. But you rely, to some extent, you do rely on the judgment of the people who are doing it. There's one exception which we learned, which is when it comes to regulatory correspondence. That was the one piece of the due diligence report that I always tried to look at the primary correspondence with the agency. Because it's the one where you actually get a sense of the nuance of the program and what the agency really thinks about the product and when
potential added value, etc.
Yeah, and so there's a big difference. I found I believe between looking at drugs that are sort of like transformational versus drugs that are me too, and those look very different when viewed through a regulator's eyes.
I like that actually. I recently talked with a friend, and we were talking about the company evaluating, and then he rapidly was an approved drug, rapidly went to see what the regulatory, what's the folder and what they really say and how they compare it to existing and what is their take, and then you feel it's very different message than on what I think was a Roche drug, but it was very different from the Roche commercial pitch, and it was very interesting to see actually, and very like detailed, but also very quite understandable from even like not like super KOL level to read it. I mean, even for me, I could read it and pretty clearly understand of how it compares, which I found very good actually. I haven't done it for Adaptimmune. I haven't checked what the FDA says, but I guess for you, it's
interesting. There's a whole load of documentation that's out there that shows the discussions that we had, and there's an interesting balancing act on this, because it was actually a privilege going through that process, because and you look at it from two angles. So we had the FDA, we had to manufacture Tecelra. So the FDA inspected our manufacturing facility at the Navy Yard, and we had an inspector there for 8 days, all of 1 week and then into the next week, and they were exceptionally thorough with what they were asking, and you've got this duality in your head going on, because on one hand it's like, we've got this, we know what we're doing. Like, there's a sort of like, okay, we want to get through this, we want to provide you with everything, but like, this is a lot. And on the other hand, there's the view of the patient, which is, okay, this is a regulatory agency that is going to give permission to a manufacturer to manufacture this product. Actually, as a patient, I walked away from that process feeling, actually, that regulator is doing their job. They are going through that process. And from the patient side, I was like, okay, if that's the process the regulators are going through, then actually I feel better as a patient on a potential society that regulator is actually regulating effectively, even though it can be, I don't think it's frustrating. It's just like, it's tense. If you're going through this inspection for the first time, it's a tense process. But from a patient perspective, it's what you anticipate the regulator should do.
It talks also even from an industry point of view. It brings trust to so many levels, and at the end of the day, that's also what you want. Global healthcare, bringing healthcare to patients. I mean,
you do,
and especially when it's complicated and the patient cannot understand.
And for these therapies in particular, I think, because they're manufactured on a patient-by-patient basis, the manufacturing is different in terms of the mechanics of it, and then the extent to which the product is the process is so much more true for a cell therapy than it is for a biopharmaceutical or obviously for a small molecule, because your ability to characterize that product and say you produce the same thing is quite limited, because the input cells are very different.
I want to talk about manufacturing. I wrote down here from a previous thing you said, but maybe to finish this on your personal background, you talked a lot about BD as well, and your BD experience, but then at Adaptimmune, on the BD, at least you went to the commercial on your own. You didn't try to partner with someone. So how intentional was it?
So that's not a discussion that says we will never partner on a global basis. In fact, we've sort of been clear. We have some fundamental platforms, we have a pipeline, we have some products. Leveraging the value of those on a global basis to the maximum extent is going to take partnerships. So very, very clear about that. Having said that,
let's get Galapagos, which we can talk a bit after.
So that's a really good example. And then what previous partnerships we've had with, for example, Genentech on our Allogeneic platform. Other examples how we can leverage the platforms that have broader utility than we can provide. If you've got these fundamental platforms. But at the same time, I think that there is a desire or a belief that in a new modality like cell therapy, the way that we've done everything up to this point in time has not been the same as it would be if we were developing a small molecule. And there's lots of things that if you're developing something that is well known and understood, it's easy to outsource or partner bits of the value creation chain. But for us, that's not as apparent in this new modality. And so the way we go to market, and the fact that so much of the commercial investment is actually on the supply chain, it's on actually how you deliver this product to patient, sort of meant that for this particular market, which is relatively small, this is sort of perfectly sized for us, and it's something that we felt not only could we bite off, but actually, if you said who's the best owner of this, it would be us, because I think we are able to best extract value from this, for deliver value to patients and extract value for our shareholders.
Yeah, and I guess you had also access to capital, as you mentioned. You had the optionality to do it on your own. There's a lot of biotechs who don't have the optionality.
Well, it's also, if you think about our commercial, the actual true commercial investment, ignore the sort of manufacturing investment and just think about the commercial investment. It's relatively small organization. Our commercial plus med affairs organization is about 30 people. We're going to be targeting about 30 sites in the United States. And so, it's discrete, but that's what's necessary. This is a rare disease white glove model as opposed to a mass market model, and so I think it actually is perfectly suited to smaller companies who are very specifically focused on delivery in this area.
It's a typical Genzyme model. I read the How Do You Tell Me Your Biography?
Yes.
Which I can recommend as a book, but it sounds like that model. Yeah, that's great. It's good. And that's a perfect transition to manufacturing. I read quite a lot about this, and I heard that several times in cell therapy. Manufacturing is the product basically, and it's key. I saw some examples even in CAR-T where the manufacturing on the same CAR-T can make a huge difference in the fitness of the cells, etc., etc. Can you maybe expand on that for the audience of how important manufacturing is, and maybe if you have some examples from Adaptimmune or not from Adaptimmune?
Yeah. Well, so I think it's fundamental, and I did not understand it when I joined the company. Although I think the people who have been deeply in cell therapy for a long time have this is what they live and breathe. Everybody talks about these things as if the primary differentiator for a therapy is the thing it targets. So we talk about CD19 CAR-Ts, and we talk about MAGE-A4 targeting TCRs or PRAME targeting TCR. And that's fine. That's the bit that we genetically edit into the cells. And therefore, it is where a chunk of the IP comes. It is what gives it the ability to recognize a tumor that it previously wasn't recognizing. But the cell is doing 150,000 things all at the same time. And one of the beauties of cell therapy, there's two things that differentiate the cell therapy in my mind. One is this one-off therapy that gives permanent gain of function. That's the genetic editing part of the cells. That's effectively gene therapy in a cellular form. But the other piece of the cell therapy, and the differentiator from gene therapy and from every other therapy, is that what you're giving is a little engine, and you can tell that little engine to do a whole load of other stuff. So you can take cells that are previously in a particular state, and you can change that state through your manufacturing process. So you change it by editing, but you change it, and I think it will be at least as much by the manufacturing process, and that's what's been demonstrated in a variety of places. Novartis's T-Charge, where they show fundamental differences in the fitness of cells. That shorter manufacturing time seems to be very significant and replicated across a range of different types of therapies. The one that we have is which we've talked about publicly is that when we moved from our Tecelra, previously at Famicell, to a product that's now called User-Cell, which is a next generation version of Tecelra. We did two things. We did a number of things to it, but two of the things we did for it is, one, we used the same T-cell receptor, but we added a CD8 to make all of the cells killer cells, and they preserve their T helper cell function, but they were all turned into killer cells. So you get a much more potent product for a particular number of cells. And then the second thing that we did was we added an AKT inhibitor into the manufacturing process. And what that was designed to do, and what it seemed to do, was enable the cells to replicate, to expand, without growing older, without differentiating as much. So you get more cells, you get a similar dose of cells, but they're not as old, they're not as exhausted, they're not as terminally differentiated. So they're not as far along their sort of program into T killer cells or into terminally differentiated effector cells as you would expect. And the advantage of that is that they go into you and they then expand more in you. They then persist for longer, and they have a more stem phenotype. They have an earlier phenotype, which means they are more capable of replicating, and they will do that in vivo as opposed to sitting in an incubator at Adaptimmune.
Okay. And this makes total sense, and translate then to efficacy and control of those.
Completely different growth curve when they get into you. So the Famicell grows very quickly, expands very quickly. You get any cytokine release, which we don't see that much of with a Famicell, quite quickly. With User-Cell, it expands much more slowly. PP expansion takes a few more days. We get delayed cytokine release syndrome to the extent that we get it, etc. So it's just a completely different phenotype of product, behaves very differently, and actually is effective in a bunch of solid tumors that a Famicell Tecelra was not effective in. This Tecelra was very effective in sarcoma. Outside of sarcoma, modestly effective. User-Cell has been shown to produce responses in a wide range of other solid tumors.
Okay, that's amazing. Very clear explanation. So, and it brings me also to another question, very similar. I mean, in a similar tone or category. I talked with a friend at J&J, and he obviously quite involved in CAR-T as well and in cell therapy. And he was curious what's your take on, let's say, the next generation or next wave of cell therapy, especially when you're talking about allogeneic or in vivo, in vivo cell therapy. What's your thoughts there?
Yeah, so we have an allogeneic platform, and we're thinking about in vivo as well, long term. I think one of the amazing things about the biotech investment environment is it's constantly looking for the next hot thing, and I think it has a tendency to underestimate the challenges and overestimate the speed at which those new next hot things can be available. And I think it's very definitely true with Allogeneic.
Allogeneic took way longer than expected. Yeah,
way longer than expected. But that doesn't mean that it won't happen. In fact, we still believe it very much will happen. But it means that it will take longer because it's not going to be the same product. And so our approach was, look, actually, autologous cell therapies work, and that because they work, it's worth trying to overcome the logistical and engineering challenges of making them available at volume at scale, which is not easy, but also not insurmountable, and requires actually bringing in skills, engineering skills, logistic skills from other parts of the healthcare ecosystem that can be very useful to us. That is a problem that is being solved in real time and will continue to. And my view is the next 10 to 15 years is autologous. Okay, so then I think you get
that point.
I think we think it's the next decade. Now, in the hem-onc space, you might get to donor-derived allogeneic before then. But in the solid tumor space, nobody's close. So I think the next 10 to 15 years is auto, allo, and then allo maybe gets overtaken by in vivo. I don't know. All of that's out there. In the meantime, there's a lot of other innovation that can be done, and there's sort of a whole bunch of scientific innovation. I think multi-targeting, multi-edited, multi-armored approaches, and finding a way of developing these rapidly, iteratively, and a regulatory environment that enables that is going to be critical to success beyond the first half dozen, 10 products that people will get on the market targeting PRAME. Because the beauty of cell therapy is you can iterate these things. And then the other piece, I think, is just on the manufacturing. When we understand all the parameters that actually define efficacy for our products over the long term, and we're sort of scratching the surface of that at the moment. When we understand that, then you'll be able to design manufacturing processes that are dramatically more efficient than the current manufacturing process. And that's the key to getting to this being in any way, a truly global, widely available option. How do you get out from the expensive medicines for the Western Hemisphere and into a global solution for a large subset of cancer? I think that's going to be key.
That's a great way to finish on the industry. Before we have just a few minutes left or 2 minutes left. Usually I finish with just rapid, a quick fire, just yes and no questions.
Go for it.
So go for it. And some are a bit more funny. The first one is funny. Do you prefer Philly cheesesteak or Oxford fish and chips?
Philly cheesesteak, I'm afraid. Sorry. Yes.
What's your favorite biotech book?
Emperor of All Maladies.
Oh, I don't know this one. I was like,
yeah. It's a biography of cancer. Wow. A historical biography of cancer. It's fascinating, amazing read.
How much do you read, actually?
I mostly read non-fiction and news. I'm not a huge fiction reader. No, so I'm.
How much?
And the answer is not as much as I should. Yeah.
What's your what's your one of your biotech heroes?
Again, with the risk of being stereotypical, because I'm sure it's on a bunch of people say it's John Maraganore, founder CEO of Alnylam, because he had an idea in a completely new modality and did absolutely what it takes to make that Alnylam the successful company it is today.
I saw he recently received a lifetime award or something.
Yeah, it's an amazing story. And there are several of them, but he's particularly inspiring.
Amazing. That's great. Let's finish here, and you're perfectly on time for your next meeting first.
Thanks,
Adrian. It was amazing.
Great to speak with you. Take care.
Yeah.
Cheers.
Bye-bye. I'm impressed by the development of Tecelra and Adaptimmune and the benefits it will have for sarcoma patients. I'm also impressed by how clearly Adrian could articulate the big picture of TCRs, cell therapy, autologous versus allogeneic versus in vivo, while having fun. If you've also enjoyed this episode, please hit the like, follow, or review button. Any of those actions would help us a lot to give more people access to the podcast. If you want to see similar videos, please check out our channel where we have many available. Also, I would be curious to hear what you think so I can further improve the content. If you could leave a comment wherever platform you are or shoot me an email at philip@flot.bio. Alright, thanks for watching to the end and see you in the next episode.